Okay, so today I'd like to discuss an article from Science Daily titled Scientists Just Discovered a Secret Code Hidden in Your DNA. It is about transposons, specifically the Mir11 family and their role in human gene regulation. Before taking this course, I thought of transposon more as a junk DNA, but the article showed otherwise. This element can influence gene expression, especially during early embryonic development. Also, MER11 elements are distributed via a copy and paste mechanism, which is directly related to how we study the duplication of genetic material. This copying can lead to the emergence of new functional regions of DNA. So I was particularly interested to learn that the researchers divided MER11 into subtypes G1 and G4 and found that the young G4 subgroup is the most active and enhances gene expression in stem cell. This gives me a real-world example of the concept of neofunctionalization we discussed. Duplicated elements or genes can acquire new function. At the same time, other subtypes lose activity over time, reminds us of pseudo-generalization. Thus, the fates of the duplicates we discussed in the course, subfunctionalization, near-functionalization, and pseudogene formation can be traced even in the case of transposon. The articles also touches on the topic of evolution and sequence conservation. Scientists compared MER11 in different primate species and identified conserved regulatory motifs. This resonates with the sliding window method we studied, which is used to find conserved regions in intergenic regions. Such regions are important because this may contain regulatory element, as if in the case with MER11. I also considered which methods from the course could be applied to a similar study. For example, to compare MER11 subtypes across species or to find their orthologs and paralogs. BLAST could be used. These tools allow us one to find similar sequence and evaluate their statistical significance using e-value. This directly relates to my new understanding of how to classify genetic elements in an evolutionary context. So I also thought about the particular analytic methods. We studied the microsatellites analysis, DNA profiling, and mini satellites through DNA fingerprints. Similar approaches also not used in the study could help classify different copies of MER11 and study their distribution among population. This gives me the opportunity to mentally connect the course experimental method with real-world problems in genomics. Ultimately, this article helped me connect the various models of the course from the mechanism of gene duplication. Sorry. Uh, so ultimately, the article helped me connect the various models of the course from the mechanism of gene duplication and evolution to the functional role of non-coding sequencing and transposon. I realized that all these processes are closely interconnected and together form a dynamic picture of the genome. For me, this reflection proved valuable. I saw that the method I'd learned BLAST, ORF searching, sliding window, paralog, and ortholog classification, and variation analysis aren't abstract concepts, but real tools that help explain recently discovered phenomena such as the role of transposon in gene regulation. Yeah,